today we will see in this video what is transgenic plant. What is a transgenic plant, or what are transgenic plants, or how we produce a transgenic plant. Okay. Transgenic plants are those whose genome is altered by adding one or more foreign genes with the help of genetic engineering technique. Okay, it helps in crop improvement. Okay, why we do that? For crop improvement purpose, like we need a better crop. Okay, then it also ensures food security. Then it helps in increasing the yield of the crop. And there are many other applications. Okay, basically this is how done. Bacteria, actually there are various methods of transfer of the genes into the plant cells. There will be biological method which include agrobacterium, mediated gene transfer. There will be plant virus vectors will be there. These are biological methods. There will be some physical methods which I have already discussed in a separate video, like your physical methods will be your electroporation, your micro-projectile, micro-injection, liposome fusion and all. I have separate videos regarding physical methods. Okay, in this video we will focus on the agrobacterium mediated gene transfer. Okay, basically how it is done. The general overview is that the bacteria Agrobacterium tumefaciens is able to transfer is plasmid, which is known as TI plasmid, inside the host cell or plant cell, which ultimately results in the formation of tumor formation, which is known as crown gall disease. Okay, so what we do, we apply this capacity of this bacteria to transfer its gene in order to transfer our gene of interest inside the cell. Okay, that means we modify the plasmid in such a way that it doesn't cause the disease, but it will be able to transfer the gene that we want to transfer to the plant so that it gets improved, and a recombinant or genetically modified plant is formed. Okay, suppose this is the bacteria I'm talking about, Agrobacterium tumefaciens. So it will have its genome, sorry about that, it will have its genome, bacteria, bacterial genome, and there will be a plasmid. Okay, suppose this is your plasmid. Then this plasmid will be isolated. Okay, and cloning will be done. Cloning means some portions of this plasmid will be cut and a new portion will be inserted inside this bacterial plasmid. Okay, if this is suppose your bacterial plasmid, which is TI plasmid, then there will be some portions, okay, which will be cut, because we need to keep the size of the plasmid same. Okay, so some portions will be cut from this, which is not required for transfer of the tDNA. With the portion of the plasmid which is transferred is known as tDNA. Okay, so some portions of this tDNA are cut and our gene of interest is inserted inside that portion. And now with the help of different enzymes or genes present in this plasmid will help the transfer of our DNA inside the plant cell. And now with the help of tissue culture technique, we can grow plants that contain the particular gene that we have inserted in order to make some improvement in the crop. Like if you want to produce an insect resistant plant, or drought tolerant plants, or maybe some plants we want to modify a plant that can give you more production of a fruit, something like that. That is why agrobacterium is treated as nature's most effective plant genetic engineer. Okay, so let us first see what the TI plasmid actually contains inside it, or what is the genomic organization of TI plasmid. Okay, the TI plasmid which we are talking about is from Agrobacterium tumefaciens. So it will have this tDNA region that will be actually transferred in order to cause crown gall disease in plant. That means what? Whenever this gene get transferred from Agrobacterium tumefacient inside the plant cell will start developing lot of tumors, and it will inhibit its growth, like its shooting and rooting and all. So let's see how it is done. Okay, this plasmid DNA will have two borders, left border and right border, which will be of 25 BP length. And inside it there will be gene for synthesis of auxin, there will be gene for synthesis of cytokine, and there will be gene for synthesis of opine. This auxin and cytokines are known for inducing tumor formation. And the opine gene produces opine that will be catabolized with the help of opine catabolizing proteins, so that the bacteria can take the carbon, nitrogen and everything, the nutrient source from opine. Okay, so whenever this bacterial tDNA get inserted in the plant cells, so the bacteria can take out nutrient source with the help of opine synthesis from the plant, and also it starts forming auxin, cytokinin, and this results in the formation of tumors inside the plant cell. Okay, so this was about the tDNA region. Now comes your virulence region. This is your virulence region. In this region, lot of genes are present. Okay, many genes are present, which will produce different proteins in cascades during infection, and then this virulence regions, actually these proteins that will be formed, are responsible for cutting and transferring the tDNA region in the single stranded form, that will be transferred in the single stranded form inside the plant cell. Okay, ultimately transported to the plant cell nucleus. And I have already talked about opine catabolism region, and there will be an origin of replication, which will be responsible for replication, okay, of the plasmid inside agrobacterium. There are basically three types of agrobacterium: Agrobacterium tumefaciens, which cause the crown gall disease. Then comes your Agrobacterium rhizogens, that cause your hairy root disease. It has RI plasmid. And your Agrobacterium radiobacter. This seems to be avirulent, doesn't cause any disease. Okay. The segment of the DNA in Ti plasmid that is actually transferred inside the plant cell is known as tDNA or transferred DNA. Okay, tDNA region. The virulence region of the Ti DNA has different genes. Okay, such as VA genes. Okay, these are all genes: VA, VG, D1, D2, E, B, 11, H, E2F. All these are responsible for contributing in tDNA transfer. Whenever the plant cell gets wounded, it releases some components like acetylserine, some sugar compounds, and some phenolic compounds other than acetylserine. This type of substances are sensed by the bacterial cells, and those acts as a signal for activation of VA. So you can say VA protein can sense acetylserine. Okay, I'll use different ink here. Sense acetylserine. Then comes your VG. What happens when VA get activated? um, it will, you know, phosphorylate VG protein. Okay, VG protein will be VG protein will be phosphorylated. Therefore, what happened? VG protein will then, activated VG will then trigger the transcription of all other genes that are present in the Vir box, like your D protein, VD, VE, VB, VH, VRAF, and all. Okay. So, what is the function of VG? It will activate the Vir box genes. There comes Vir D1. It will excise the tDNA at borders. Okay, at only one strand. Excise the DNA at only one strand, because it will be transferred as a single stranded structure. And VD2, it protects the 5' end from being cleaved by exonuclease. Protects 5' end from exonuclease. Then VE helps in the gene transfer by covering the DNA inside the plant cell. Okay, it will helps in gene transfer. Then VB forms conjugational form. So, for transfer of the DNA from plant to animal requires some, it will require a road. Okay, how this road is formed? By formation of conjugation pore by VB, it will form a complex like structure like this between the bacterial plasma membrane and plant cell membrane, and through this pore, the DNA will get transferred from bacteria to the plant cell. So, form conjugational pore. Then Vir B11, it has ATPase activity that provides energy for movement of DNA. Energy for movement. Then comes your VH that detoxify other adversely affecting compounds during the DNA transfer. Then VE2 forms a channel in the membrane. Okay. VRAF, the actual function of VRAF is not understood, but it probably assists in the transformation of normal plant cells to tumor cells. So, if you see in this diagram, this has been taken from Google. Okay, so you will get it if you search it. You can find it in Google images. So this structure shows that suppose your plant cells get wounded somewhere, then it will release some phenolic compounds that will be sensed by the various proteins present in the plasma membrane of the bacterial cell, and it will activate Vira proteins of the virulent gene. Then Vira protein will phosphorylate VG that will activate the cascades of protein formation from the other Vir genes. Then your VD one will excise the tDNA here, and it will take out a single tDNA from left border to right border. That will be then transported, and during transportation VD2 will bind to the 5' end in order to provide protection from exonuclease breakage. Then this will be transported through these channels, that is the conjugation pores formed between bacteria and plant cells, which is formed by Vir-B complexes. Then it will move through this pores, and then what will happen inside the plant cells, Vir E protein will bind with the DNA and it will form a covering structure like this. Okay, so that the DNA does not get cleaved inside the plant cell cytoplasm, because there will be a lot of endonuclease enzyme will be present, endonuclease, exonuclease both. So if the DNA get covered with different enzymes, the endonuclease enzymes or exonuclease enzyme would not be able to recognize the DNA. Then what will happen? It will be transported, when it comes near the nucleus, it will be transported inside the nucleus with the help of NLS protein, that is nucleus localization signals, and then it will be when it will be inside the nucleus, it will form double stranded structure with the help of some other proteins like VC, and then this double stranded structure will get incorporated into the plant cell genome. So now the part of the gene that get incorporated will have the gene of our interest, suppose antibiotic resistant gene or insect resistant gene, whatever. So it will have that particular gene that we actually wanted to get transferred inside the plant cell. So this is the overview of how Agrobacterium tumefaciens can transfer our gene of interest inside the plant cell. This is actually this plasmid DNA that we are going to use for transfer of our gene of interest will not contain the genes for formation of auxin and cytokines. Okay, these are called oncogenes because they are responsible for formation of the tumor. So the plasmid that we are using will be a recombinant one that will contain the gene of our interest and only the virulent gene. Okay, that will be responsible for transfer of the DNA.